The deep ocean serves as an incredibly vast, active pharmacological reserve. The initial proof of concept happened in the 1950s. Researchers examining a highly stationary Caribbean sponge discovered unusual nucleosides, compounds that structurally mimic the building blocks of DNA and RNA. Chemists successfully isolated and synthesized those nucleosides to create cytarabine, a drug that directly disrupts DNA replication in leukemia cells, and vidarabine, which targets severe viral infections. These two drugs proved that marine chemistry could be safely deployed in human biology. From there, researchers shifted their focus from static sponges to active predators. The cone snail is incredibly slow, so it relies on a highly specialized venom to instantly paralyze fast-moving fish. Scientists successfully isolated the active peptide in that venom to develop ziconotide, known commercially as Prialt. Unlike traditional pain management drugs, ziconotide does not interact with the brain's opioid receptors, meaning it sidesteps the risks of systemic tolerance and addiction. Instead, it is delivered directly to the human spinal cord. As this visualization shows, the peptide acts as a physical gatekeeper. It specifically targets and blocks N-type calcium channels at the synapse, stopping severe pain signals before they can travel up the nervous system. By understanding the precise mechanism of the toxin, researchers successfully re-engineered a lethal marine defense into a highly effective tool for chronic pain management. In the field of oncology, the focus is on forcing abnormal cells to self-destruct. Compounds extracted from sea squirts, specifically trabectedin and plitidepsin, achieve this by binding directly to minor grooves in DNA, which actively triggers programmed cell death. Delivering these highly potent toxins without harming healthy tissue requires extreme precision. That's where compounds derived from the sea hare come in, laying the biological groundwork for antibody-drug conjugates, or ADCs. An ADC operates exactly like a microscopic homing missile. The antibody navigates the bloodstream, locking onto a receptor unique to a Hodgkin lymphoma cell. Once attached, it safely injects its payload of marine-derived cytotoxins directly inside the tumor. The ecological math of scaling these treatments presents a major hurdle. We cannot dredge millions of wild marine invertebrates from the seafloor to meet global pharmaceutical demand. The solution requires laboratory replication. The chemical structure of Helicandrin B, found in a Japanese marine sponge, is staggeringly complex. Yet chemists managed to completely reverse engineer and synthesize it in the lab, creating the breast cancer drug eribillin. Achieving total synthesis means we can mass-produce these intricate marine compounds on demand, leaving the fragile deep-sea ecosystems entirely undisturbed. This chemical pipeline extends far beyond specialized oncology. It forms the bedrock of daily preventative health, specifically marine omega-3 fatty acids. There is a widespread misconception that fish naturally produce EPA and DHA. They do not. These highly concentrated lipids originate much further down the food chain, synthesized primarily by marine microalgae and accumulated by small forage fish. Looking closely at this microscopic sample, you can see the dense internal pigmentation of the algae clusters. These single-celled organisms serve as the original biological factories, producing the purest form of these essential fatty acids. When ingested, these lipids incorporate directly into cell membranes. Clinical data clearly shows they actively lower triglycerides, improve vascular function, and rapidly downregulate systemic inflammation across the human body. We have mapped and tested a fraction of a single % of the ocean's biological catalog. Now, deep-sea exploration paired with AI-driven chemical synthesis is radically accelerating how fast we can identify and sequence new compounds. The success of the marine pipeline demonstrates that the chemical blueprints needed to treat our most stubborn, complex diseases already exist in the deep ocean, waiting for us to find and translate them.